So let's talk about the genomic side of long COVID now and what we can do with genetics to help understand disease mechanisms. So first of all, when this COVID pandemic began, it quite quickly became clear that COVID was a severe or even lethal disease for some, but quite mild for others. And there are, of course, different kinds of risk factors that affect these individual differences, such as obesity or age and so on. But also genetic factors that we inherit from our parents can play a role in these individual differences. So scientists from the FIM, so the Finnish Institute for Molecular Medicine at the University of Helsinki, where I also work, put together this COVID host genetics initiative, which is a global collaboration to study how these genetic factors also might play a role in the individual differences of COVID. And it quite quickly grew to global scale with tens of studies around the world joining the work. And they have found then tens of genetic risk factors that associate to susceptibility to the SARS-CoV-2 virus, so how easy a person actually gets infected when they are close to the virus, and how then the severity varies between individuals. So that tells us a bit also how the inherited factors play a role there in why some people have just a cough and some suffer a very severe infection acutely. But there are also differences in the recovery time, and as we know, many people actually suffer for months or even years. So how about genetics then playing a role in this? Well, first of all, I would like to still take one step back, just reminding that COVID or SARS-CoV-2 is not the first infection known to cause long-term health problems. So long COVID, of course, became known to many around the world media and so on, because so many people got this new virus in a short period of time around the world. But infections and other pathogens like virus or bacteria are known to cause diseases via various mechanisms. So for example, it can be a resident virus in the body that can activate years or even decades after the acute first disease and cause diseases like chickenpox, the virus varicella zoster, causing shingles, or then infection can trigger an autoimmune disease, like in the case of Epstein-Barr virus that Talia also mentioned there. So that can affect then, for example, multiple sclerosis. Or in the swine flu or the pandemics vaccine that was used for that influenza virus, there were also these autoimmune processes affecting the risk for narcolepsy, especially in children. Then also viruses like HIV or human papillomavirus can increase cancers by suppressing normal immune responses or other molecular disease or molecular defense mechanisms. And SARS-CoV-2 is definitely not also the first virus showing to cause post-viral fatigue and other symptoms. But how can then how do we understand these mechanisms in long COVID? So various mechanisms have been already suggested, as you know, and you can see here some of those immune system related ones from the previous slide and the other diseases. So for example, we can have lingering SARS-CoV-2 virus, so infection in some tissues that can then cause symptoms. It can be also a reactivation of other viruses like the EBV, as heard in the previous talk. or the varicella chickenpox virus also. There have been shown some autoimmune mechanisms in long COVID as well. And then there can be other mechanisms also, like tissue destruction during the acute disease, especially, for example, in the lungs, but also other tissues in the body. There can be microglottin in the blood stream and dysregulated blood coagulation that can cause various symptoms, also heart symptoms. And then there can also be, of course, we have to remember, like when people are treated in the hospital, especially in the intensive care, it takes time to recover. But also there can be neuronal signaling differences. For example, in the brainstem that controls breathing, heart rate, and alertness. And that can potentially lead to symptoms like fatigue, brain fog, sleep disturbances, and the breathing problems, and also dizziness when one is standing up. Also some neural dysfunctional signaling in the vagus nerve can affect heart rate and digestion and immune responses. But how can omics research help understand this complex puzzle and then actually help the patient at the end? So what is omics or genomics? If we think of genomics, so the genes as the DNA molecules stored in the DNA molecules in the chromosomes in each of our cells, those genes are kind of recipes. So it's a recipe book, how to make then the proteins that we need. And in between there is transcriptomics, which is then if we have a gene, then that gene is expressed into messenger RNA, and that can then make a protein. So kind of baking the cake here using that recipe, and that those proteins that then are part of our metabolism and also part of the structures that we build in our body and kind of things that happen. And then we can study these different parts of this system using the different omics, and we can combine this information. But how does this help actually treat the patients? So first of all, if we find a genetic difference, for example, between patients, long COVID patients and control individuals, it can be in a gene that then codes a protein, or in a regulatory region in the genome, controlling, for example, the expression of certain gene. And this can tell us about the biological pathways and the functions and disease mechanisms, which can then help us and other researchers develop new drugs, and also kind of a fast lane for treatments, find drugs already used for other diseases that can be repurposed and like used for this new one. Because they are already kind of checked that they are more or less safe to use and so on. So that can be a fast lane to find some treatments for long COVID as well. Then we can have like more personalized medicine, where we can see like which parts of the patients have this kind of these kind of mechanisms in their disease, and then that can then reduce the adverse effects of certain drugs and so on. And it's also possible to find biomarkers to find risk groups, for example, the BRCA genes in breast cancer, and then develop diagnostic tests. So what are we trying to do here? We are trying to identify genetic factors that either increase or decrease the risk for long COVID. And we're interested if these are independent of susceptibility to the acute infection, and what about the severity of the acute disease, and are these specific for this virus or more general post-viral, and how do other diseases then are they similar? So we have started this long COVID host genetics initiative, and we just published a couple of days ago a paper using the data from 24 studies from 16 countries around the world, around 6,000 long COVID patients and 40,000 COVID positive controls without long COVID, and over 1 million individuals. And actually for the replication, so to see if these were kind of correct, these findings, we even have now over 30 studies from almost 20 countries in the world, and a couple of million individuals in total. And they come from different areas around the world, mostly Europe and European ancestry from the US, but we also have some data sets from other countries. And what we do with this data is that we compare those who have long COVID to those who have had COVID but recovered within 3 months. So it is quite a long period already to suffer from an infection for almost 3 months, but still that is the cutoff there. And then we compare these long COVID patients to general population, so everyone else in each data set that there is data from. But how to define long COVID? As we heard already, it's quite complex, or even COVID actually in some cases. So we can either look at electronic health record data, so diagnoses that people have in their health records. And there is a specific code for long COVID called post-COVID condition, and then also code for the SARS-CoV-2 infection, whether it's been verified by a test or not. And then there's a second approach to ask people, well, what kind of symptoms you had and how did you recover from them? So we have also data from questionnaires from some of our data sets, and there we mostly use the World Health Organization definition for long COVID or post-COVID condition. So it's the symptoms that occur after a probable or confirmed SARS-CoV-2 infection, usually 3 months from the onset of COVID, and that last at least 2 months and cannot be explained by an alternative diagnosis. And the symptoms we see vary between the different cohorts around the world, but the most common complaint is fatigue. Then there are loss of smell, which is a bit less common in the later phases of the pandemic, and shortness of breath, cognitive dysfunctions, different kinds of pain, sleep problems, and so on. And what we do then, so we define the long COVID cases and then the controls with COVID or just everyone. And we take blood samples and extract the DNA, so the genetic material inherited from parents. And then we compare the long COVID cases to the controls. And we get a sort of a Manhattan plot. So I'll show you a real Manhattan plot soon, but this is it's kind of a plot where we have all the chromosomes on the x-axis, and then we have kind of peaks so in the Manhattan skyline showing which parts of the chromosome or the whole genome is kind of more associated to this disease, in this case long COVID. And we take into consideration the patients or the people's age and sex and so on. And then we get these results, and we can also then use them in a meta-analysis, so we get data from different studies all together. And when I've done that and looked at long COVID after test verified SARS-CoV-2, and this was the paper published on Wednesday this week, so it's now online. We see this peak on a Manhattan skyline, and here are the chromosomes. So this is the chromosome 6, and then the higher the peak, the more significantly it is associated, this area in the genome, to long COVID. And we see that there is this is kind of an address for the genetic difference, the long number there, and then most people have a G, and then more of the people who have C have long COVID, so they have 60% higher risk for getting long COVID. And this is how it looks then in the kind of DNA, that most people have the G allele, and then those people who have long COVID, they can have more of them have the C, but it does not mean that anyone who has this one difference in one spot in the genome would then get long COVID. So of course this genetic difference affects like the long COVID risk together with other genetic factors and also lifestyle factors and environment. And what does this mean? From earlier studies, we have seen that this area there is a FOXP4 gene, and it is a transcription factor that works in the lungs and also other tissues and controls lung functions in health and disease. So it can be associated with asthma and some lung cancers, but also severity of the acute COVID. And one interesting thing is that when we look at this specific variant across the different studies that took part in this, and when we look at how many people actually in those populations have this risk allele or risk variant, actually it's not that common in basic European population. It's about 2% of the people carry this risk factor, but it's more common in Finns because we have a bit different genetic or genomes in some spots. So 14 people around in Finland carry this, and this does not mean again that 14% of the people here in Finland. So not 14% will get long COVID, but they have a bit higher risk for it. And then in some populations, even 1/3 or half of the individuals can carry this risk. Then there are some other studies also of the genetics of long COVID. This is now a preprint from the 23andMe, which you might have heard. So they collect people's blood samples and run like commercially these genetic analyses. And then they also can use people's data for scientific studies. And they have pre-published this study, so this has not been peer-reviewed, so it has to be like taken a bit with a grain of salt. But they have found that people with long COVID have, or like people with this difference in the HLA, so human leukocyte antigen area in the chromosome, also 6, but different area, have a bit higher risk for long COVID. And this area is connected to adaptive immune system and how viruses are presented on the immune system cells. And then they have also seen a lower risk for those with blood type O for long COVID. And then there was a recent study this month also from a population sample in Germany, which found genes that work in the immune system, for example, activation of macrophages, one type of immune system cells, and then also genes in the repressing viruses or releasing virus particles from cells that are infected with the virus. And they also found a difference in olfactory receptor gene, which makes sense in the case of loss of the sense of smell or taste. And in addition to these specific sites in the genome, these genome-wide association studies and genetic and genomic studies can tell us how different diseases are connected to other diseases. And we can run this kind of genetic correlation to see if, for example, long COVID is similar with some other diseases like chronic pain or chronic fatigue, fibromyalgia, different kind of autoimmune diseases, anxiety and depression, and so on. And this can again then tell us more about the disease mechanisms and also help find treatments for these different diseases that can still have some genetic similarities. But what are we doing now with this this study together with the EU project with Helsinki University Hospital and the other partners? So when we have genetic data and then and patient data or some way to kind of look at the cases and controls, we can find these genetic risk factors, and then to understand how those actually help the patients in the end or what those mean, we can combine that with publicly available functional data, for example, how those genes are expressed and used. So how the recipe book then goes into the production of or cooking that cake, for example, and so on. But what has been often missing is like this functional data that would be measured in patient populations, because often that is from more or less healthy individuals. And also then using this like patient population information or patient data with controls from the same area, for example. And this makes this EU project with HUS and Basel University and Lifelines and all these other partners studying the different omics and different parts very important. So some take-home messages. So this FOXP4 locus in the chromosome 6 associates with long COVID. So it increases the risk for long COVID together with other genetic and lifestyle factors. And it's connected with lung functions. And then there have been other genes, of course, also from other studies that either increase or decrease the risk for long COVID. And most of these are associated with the regulation of the immune system, and some for blood groups and olfactory receptors. And these kind of global genomics collaboration studies, especially with diverse ancestries, can help reveal mechanisms of diseases and thus then inform how to find new drugs or use drugs from other diseases. And also studying COVID-19, we can understand also other post-viral and fatigue-related conditions and prepare for future pandemics, which I hope we won't have, but of course good to prepare. And I would like to finish by thanking everyone around the world who has made this collaboration possible. So this is from the Long COVID Host Genetics Initiative, led by me and Hanna Ollila from the FIM at University of Helsinki. And then here with this Long COVID EU Horizon project, we have worked with Lea at Hanna's group, and also Maija Puhka and Päivi Lahermo from the FIM. And then I really want to thank all the partners who are leading this project and working together with us. And thanks everyone who is still here listening to this. I know it's quite heavy and a lot of information at once from the three speakers, but thank you all.